The following slide and video show presents step by step how to perform malaria microscopy. We start with the preparation of the slides, which means actually to prepare the thin and thick films for malaria microscopy. This might be a bit different than are you used to in a diagnostic context, but in a research context, it is very important that we prepare the slides in a very standardized way, meaning we actually use defined volumes for both the thick and the thin film.
I'm placing a drop of the blood here for the thin film and then 3 drops of the blood for the thick film. I'm using the glass slide as a spreader to prepare the thin film. At an angle of 45 degrees, you prepare the thin film. And then you use the edge of the same slide to make the thick film.
You might be familiar with the staining procedures in a diagnostic context. In such a context, you want to have a result very quickly because you want to inform the patient whether he or she has malaria or not and initiate the treatment as soon as possible. So you apply the so-called fast stain using a highly concentrated Giemsa working solution and stain for a short period of time. In a research context, however, you use a slightly different procedure. We do this because for us it's important to have very accurate parasite counts. So we use a lower concentration of a Giemsa working solution and stain for a longer time. We use a 3% Giemsa solution and stain the slides for 40 to 50 minutes.
We are going to prepare working Giemsa stain. You will take 10 milliliters of the stock Giemsa stain and then you add 90 milliliters of buffered distilled water to make a total volume of 100 milliliters. This is a 50 ml serological pipette, so I'll pipette 50 initially and then add 40 ml. This comes to a 1 in 10 dilution of the stock Giemsa stain. We now have the working Giemsa stain, which I am going to pour into the coupling jar that we'll be using to stain the thick and thin blood film. I am going to fix the thin blood films in methanol for 4 to 5 seconds. Fixing the second slide also for 4 to 5 seconds. I am now going to hemoglobinization of the thick blood film, that is, immersing it in water to dehemoglobinize the red cells. I will now immerse it in the 1:10 working Giemsa stain. And we'll stain it for 20 minutes. At the end of the 20 minutes staining period in the working Giemsa stain, by means of a forceps, remove the stain slide and transfer it into a coupling jar containing water for rinsing the slide. Rinse it for 30 seconds and then remove it from the rinsing water and then send it to the drying rack for it to air dry. This is the binocular microscope. It is a light microscope for examining the blood films to detect malaria parasites in the film. It has a lens system, the head, which is usually inclined. We have the body, we have the stage, the diaphragm, and then the lens system. In order to be able to see the parasite very well, you need to clean the lens system so that you'll be able to see the parasite well. After cleaning the lens system, you get your slide, you place a drop of oil emulsion on the thick film as well as the thin film. And then you place it on the stage, put the oil emulsion lens in place, and then focus initially where the oil touches the ×100 objectives. From there, you start looking out for the malaria parasites.
Before you start to count the parasites, you have to make sure that your slide is of good quality. We have different assessment criteria for the thick and the thin film. On a thick film, you should see approximately 6 to 14 white blood cells per high power field. The background is pale blue grayish. This represents the lysed red blood cells, and the slide should be free of dust and other artifacts, such as staining crystals. White blood cell nuclei should be stained deep purple, and platelets should be clearly visible and bright pink. On the thin film, at the end of the film, the so-called tail or feathered edge, The red blood cells should be distributed evenly and possibly with no or at least only very few overlapping cells. The white blood cells are stained grey-pink. The plasmodium nuclei should be stained deep purple and the cytoplasma pale blue. Hemozoin is visible as brown yellowish patches. In your context, which is mainly a diagnostic context, you may be very familiar with the WHO standard operating procedures for how parasites are counted and recorded. In a research context, however, we count and record parasite counts slightly differently. In a first step, you scan 10 high-power fields on the thick film. This is to confirm the slide quality according to the criteria that have been outlined in the previous slide and to check the overall parasite density. If you see 40 or more parasites per high-power field, the parasite count is performed on the thin film. If you see less than 40 parasites per high-power field, the parasite count is performed on the thick film. On the thin film, you count up to 2,000 red blood cells. If you can't find any parasites, you do the parasite count on the thick film. If you find infected red blood cells, you record the parasite count as infected red blood cells per 2,000 red blood cells. You also record parasite species and whether gametocytes are present or not. On the thick film, you first examine 50 high-power fields. If you can't find any parasites or gametocytes, you examine up to 200 high-power fields. If you still can't find any parasites or have seen gametocytes, you declare the slide as negative. If you do see parasites and/or gametocytes, you record the results as number of parasites per 500 white blood cells. You also record the parasite species and whether you have seen gametocytes or not. For both the thin film and the thick film counts, if you have a mixed species infection, you don't count them separately. You give the overall parasite count.